Hello again. I'm Chaitan Khosla from Stanford University, and welcome to the third part of this trilogy of lectures on polyketide antibiotic biosynthesis on assembly lines. So in the first lecture, I introduced you to the basics of what an assembly line is, what's the chemistry that happens on an assembly line, and what an assembly line looks like. We looked at the basic architecture of the 6-deoxyerythronolide B synthase, or DEBS, which I have used subsequently to make most of the points that I have made. I introduced to you the architecture of this assembly line, or at least as best as we can gauge today based on data we have. In the second lecture, I introduced you to the tools and how we've used those tools to study different kinds of questions. So we talked about refactoring a metabolic pathway like this assembly line of DEBS in E. coli. We talked about how to reconstitute that pathway in a test tube. I pointed out that you can isolate individual reactions on the assembly line and look at their kinetics, whether you're looking at extender unit selection, translocation, elongation, modification events. We also talked about how these tools can be used to study specificity, and in particular, we talked about two types of specificities. Stereospecificity. I mentioned that stereochemistry is controlled by reductive domains, as well as extender unit specificity. And I mentioned over there, you not only have enzyme substrate recognition, but also protein-protein recognition. This final lecture focuses on what, in my mind, is the grandest challenge of all in this field: understanding how an assembly line actually comes about. What do I mean by that? I've been showing you quite freely, I might add, schemes like this throughout my earlier two lectures. Every time I show you one of these schemes, or for that matter, you could open your favorite journal of biochemistry today, and chances are in any given issue, you'll find somebody having published yet another one of these kinds of assembly lines that they have cloned, sequenced, and started to characterize. And in all cases, you'll find cartoons like the kind you see in this scheme. The question that lies ahead of us is, how do we know that this is how the assembly line works? And one way to recognize why that question is not an obvious question is by thinking back to all those orphan polyketide assembly lines that I talked to you about at the beginning of my first lecture. So I mentioned to you there's these large numbers of orphan assembly lines that make polyketides whose structure we don't know. If I gave you the sequence of one of those assembly lines and asked you to draw me an equivalent scheme like the kind you see in front of you in this slide, chances are you won't be able to do it with any confidence. And the reason why I can show you this scheme in this slide and hold my head up high is because I know the answer for this assembly line. I know the structure of 6-deoxyerythronolide B, and using that knowledge, I am essentially back programming on this assembly line what must happen at each stage in order to get that final product. But that begs the question, how does nature know that this is how the assembly line must work? Why does nature start here and move here? Or putting it differently, why can't nature take a product from this module and instead of passing it on to this module, Why can't it just skip these modules altogether and pass it on to this? Or why can't it take this? Why can't it take this precursor and pass it to some other polyketide synthase that's floating around in the same cell? Where are the rules that dictate how this vectorial translocation of polyketides must occur in the manner that I tell you it's occurring in this scheme? That is the fundamental challenge associated with this field. The way we think about this challenge goes back to the work of an inspirational enzymologist from a previous generation whose name you may have heard, Bill Jenks or William P. Jenks. Jenks made huge contributions to our understanding of how enzymes work. One of the problems he talked about and gave a lot of thought to, as is discussed in this article that I would encourage any student who's interested in machines in biology to read, was the question of how do you get vectorial processes occurring in nature off the back of proteins like enzymes. This article is chock full of many really interesting insights that were way ahead of their time. I certainly don't have the time to go into all of them. But there's one insight that I'm going to focus on because I think it does a very nice job of framing the understanding of the vectorial chemistry that I'm here to tell you about. What Jenks says in this article is that proteins that do coupled vectorial processes, differ from your garden variety enzyme in the sense that they undergo large changes in substrate specificity at different points in their overall catalytic cycle. And a really good example that he uses in his quintessential chicken scratch cartoon manner that's shown over here is the example of how myosin moves on actin fibers that you probably know about because it's what textbooks are made of, biochemistry textbooks are made of. What you know is that myosin moves along an actin highway that is made up of actin monomers by essentially going from one monomer of actin to the next and so on. And that movement is powered by the hydrolysis of ATP by myosin. And what Jenks postulated back then, or what he articulated in this paper, was that the reason you get this vectorial movement is because there are different states of myosin at different points in the overall cycle. And some of these states bind tightly to myosin, the unbound states, whereas ATP or ADP bound states, the myosin does not bind very tightly to actin. The key point that Jenks made is the reason this is a machine and it's not like myosin dances back and forth on this actin highway. The reason it motors along this actin highway is because there is a distinct change in specificity of myosin for actin at different points in the catalytic cycle. When myosin binds to ATP, it exchanges with a state, unbound state that binds to one monomer of actin, whereas when that ATP is hydrolyzed, that myosin sets up the binding of a distinct conformational state to the next. It can't, this exchange between the bound and the unbound state is prohibited, because if this happened, you would be wasting this ATP hydrolysis and you'd be back to where you started from. But the reason this moves forward is because this state has a distinct specificity than this state of the enzyme. So the big changes in the specificity of myosin are what Jenks proposed is the basis for this vectorial process. We think about our challenge very much analogous to how Jenks proposed how myosin works on actin. And more specifically, there are two postulates that we draw upon from Jenks's analogy to actin and myosin to explain how this vectorial process happens. The first one is that the specificity change that Jenks talked about in our context happens at the level of protein-protein interactions. I gave you an example in my previous lecture of an acyltransferase that has protein-protein specificity in addition to its specificity for its normal substrate. I'll be elaborating on this theme much more in this lecture. And the second hypothesis that helps us think about assembly line vectorial biochemistry is that this core of the protein that exists in each module, this Ks and At, has specificity for the acyl carrier protein in an interesting way that leads to establishing directionality. Bear with me if that's not clear. Let me first start by giving you some information, some background on the relevance of protein-protein interactions. So about 15 years ago, when Rajesh Gokhale was in my lab, he came up with an interesting postulate about how assembly line directionality happens. He hypothesized that specificity that leads to this chain moving from this module to this module is not in the active sites for different substrates, but lies at the interface between these modules. So you have these docking sites that exist between modules that are orthogonal to the core modules that are doing the actual enzymology, that allow two modules to come together so that you have this directionality that is established. How do we know that? We know that through experiments like this. This is one example of an experiment that Stuart Tsuji did in my lab some years ago, where he kinetically isolated from the entire DEBS assembly line you see over here these two modules, module 2 and module 3. He pulled them out of the overall system, put them into a test tube, and reconstituted their full enzymology that's shown in this scheme, measuring the appropriate parameters that one gets from these kinds of experiments. Now what he does is he switches these short peptide linkers that flank these modules for their counterparts from this module. And if he switches just one of the two linkers, what he gets are two modules that by themselves are just as good catalysts as they were here. But when you put them together, they are simply unable to talk to each other. If, however, you take this module that has this linker instead of this linker and couple it to this module that has this linker instead of this linker, this pair over here is indistinguishable from this pair. And so now you see how these matching docking sites provide for the ability for module 2 to talk to module 3 as opposed to module 5 or any of the other modules. So you're seeing the establishing of directionality off the back of protein-protein interactions. Now what the Gokle-Suji experiments do is they tell us why module 2 talks to module 3. What they don't tell us is why don't chains go back and forth? It's analogous to the question that Jenks was asking, why is it that myosin moves in only one direction on actin as opposed to just going back and forth, back and forth? That question has an exact analogy over here because as you'll remember from the first lecture of my 3 lectures, I introduced to you that chain translocation. When a chain moves from one module to a next, it does so by a thiol to a thioester exchange reaction. And that reaction can just as easily run in the backward direction as it can run in the forward direction. And so what precludes chains from just going back and forth and messing up the chemistry so you'll never get an antibiotic out of these assembly lines? What's written in this code that allows that to happen? So the question is, how does this event lead to a movement here? But when it comes time to move the chain forward, it doesn't just go back into the same module, but rather goes to the next module. Again, over here, protein-protein interactions play a very important role. And in particular, the interactions, not just in these docking sites that Gokle and Suji identified, but also between this protein and this protein, play a very critical role. How do we know that? Through experiments like this that Nick Wu did, where he was isolated kinetically this chain translocation event, exposed this module over here to the same substrate, same docking partners, same receiving module, but now he changes the identity of the donor carrier protein. So all that's being changed over here is the identity of this protein. And what you see is you have a big difference in specificity constants just based upon who this player is in the overall chain translocation event, suggesting that these protein-protein interactions are very important. We now know through work of Chaitan Khosla and others in my lab what the basis for this is. So it turns out that these carrier proteins, which are helical proteins, have different helices, different portions of this helical protein that dictate specificity at different events. How do we know that? So you can take this red carrier protein that talks to this red acceptor module, replace it with the green carrier protein. It doesn't do a good job of talking to this acceptor module. But now if you just put a portion of this helix into this protein, now this chimeric protein is just as good or almost as good as this red carrier protein in delivering cargo through this chain translocation event, suggesting that it's this helix over here that plays a very important role in dictating the specificity during chain translocation. You can do exactly analogous experiments in that other theoretical reaction, the chain elongation reaction, where that carbon-carbon bond is formed between the extender unit and the growing polyketide chain to give you this newly elongated chain. You can kinetically isolate this event. You can use the same substrates, same catalysts, just change the identity of this carrier protein. And what you see is, depending on which module you use, you can have big effects on the specificity constant over here, as Alice Chen showed in these experiments. You can use the same kind of approaches that Chaitan Khosla used to make chimeric carrier proteins and show which portions of these carrier proteins play a critical role in dictating this specificity that is measured in these parameters. And so the take-home message from experiments like this is that even though chain elongation and chain translocation are sequential events along the assembly line. And even though all of those events involve ketosynthase acyl carrier protein interactions, the specificity of those events is being driven by different phases at this carrier protein. So this blue phase of this carrier protein is what's driving the translocation reaction, whereas the yellow phase of the carrier protein is what's driving the elongation reaction. So you have this Janus-like biochemistry in these carrier proteins. And the reason that allows you to do things in a vectorial manner is because now you can set up so that a carrier protein can only recognize the next module. How do we know that? We know that through experiments where we engineer these carrier proteins to be confused carrier proteins. This is one example of that experiment where we engineer a module that stutters. So if you take a natural module like this one shown over here and its own carrier protein over here, you give it a nucleophilic substrate and electrophilic growing polyketide chain, and you ask it to catalyze chain elongation. It'll do so. It'll do one round of chain elongation, and it'll stop at this point. And the reason it stops at this point and doesn't go back to the same carrier protein is that this carrier protein is a partner of this ketosynthase in this chain elongation reaction that forms this critical carbon-carbon bond, but it is not a partner for chain translocation. Its chain translocation partner is elsewhere on that assembly line. And so the way you can test that is you can take the recognition features of the next carrier protein, build them into this system to generate a chimeric carrier protein that can not only do a chain elongation event, but this chimeric thing can then donate it back to this ketosynthase and go through yet another round of chain elongation to give you a longer module. So here you've got a module that is stuttering, that's doing 2 rounds of elongation instead of 1 and only 1 round because we've kind of scrambled up its recognition features. And so what that allows me to do is to leave you with a basic picture of how you might want to think about this assembly line. So I showed you this cartoon. The question we're asking is, how does the growing polyketide chain pick one and only one path? It's going through at least 300 angstroms of distance as it travels through these modules from one active site to another and comes out as a 6-deoxyerythronolide B. How does it pick one and only one path to take through this entire assembly line? And so if we isolate just these 2 modules, modules 2 and 3, I can show you what I think might be a model for how this works. So we start off with the upstream module, this pink module, bound to its carrier protein in the elongation state. What Jenks would call has specificity for the elongation state. And that specificity is derived from this yellow portion of the carrier protein that docks into this active site and puts its substrate where it belongs. Meanwhile, the green carrier protein of this green module is just sitting in neutral mode. So this carrier protein then catalyzes chain elongation. It yanks out that chain. It can't return it back to that same site because it doesn't have the appropriate recognition features. Instead, it's got recognition features now using that panhandle linker, that docking site that was identified, as well as this helix that Kapoor identified. It docks into this module, but in a different orientation, so now it can translocate the chain to the next module. When that happens, that carrier protein goes back into the neutral mode, and this carrier protein, which is charged with the extender unit, comes in to do the elongation reaction. And so this 3-stroke process goes through again and again to give you the final product, to allow chains to move forward through the assembly line. And so I'll leave you with this picture. I started with the picture to the left to motivate these remarkable antibiotic machines. I've added one more picture that I suspect most of you who've had a class in biochemistry recognize. It is the picture that we often use to describe how proteins are made on ribosomes. Let me ask you the following question. What do these pictures mean to you? Chances are, if I just flash both these pictures in front of you, you would immediately recognize what I'm saying. If, on the other hand, you had just arrived today from Mars, and I showed you these pictures, they would mean absolutely nothing to you. So why do these pictures mean so much to you? I would submit to you they mean a lot to you because you have seen the movie in each case. And that is where I will leave you as the future of my field. What I showed you in the past 3 slides were essentially the first 3 frames of a movie that will be the DEBS movie over the next decade. There's no Hollywood, no Disney as yet, but it's backed up by data based upon, through a combination of biochemistry, structural biology, and similar types of chemical biological approaches. Our challenge for this field is to take those static pictures that I've been showing you so far and convert them into a movie. So once you've seen that movie, you will recognize the engineering implications of every state in every one of those thousands of assembly lines that nature has given you, and be able to rationally think about how to recombine them or how to extract interesting antibiotics from the right kind of assembly line in nature. I can't do that today, nor can any of my coworkers. But I hope this inspires you to think about how that might be done in the future. This brings me to the end of the lecture. I'd like to thank you for your attention. I'd like to thank all of my students and postdoctoral fellows who over the years have worked with me on DEBS and related polyketide assembly lines. Their names show up to the left of the slide, and I've been acknowledging their papers as we've gone along through these different modules. And I'd like to also thank my collaborators, David Kane at Brown University, with whom I've had a long-standing collaboration on the study of DEBS, as well as my collaborators at the synchrotron, SLAC synchrotron, Matthew Zirimpan, Tsutomu Matsui, and Thomas Weiss, with whom we've had very productive collaborations associated with the crystallographic and Sachs characterization of DEBS. And last but not least, I'd like to thank the NIH for having supported all our work on DEBS over the past 15 years. Thank you.